Palmiter and Brinter attempted in the formation of transgenic animals by using. Now Palmiter and Brinter taken the gene responsible for the growth hormone. The growth hormone is also called somatotropic. Now you observe gene for somatostatin, gene for insulin, gene for somatotropic. Somatotropic hormone is also called the growth hormone. Palmiter and Brinter taken the gene responsible for growth hormone. It is also called somatotropic hormone. So the answer is, next question, the assessment of characters of parent by studying the characters of its offspring is called. Now as we observe the sex limited genes, what are the examples for the sex limited genes? One example is the milk producing genes. Another example is the egg laying genes. Okay, the sex limited genes present on autosomes. The sex limited genes present in male and female. Now in our body also milk producing genes are present. In the female body also milk producing genes are present. It means we are having sex limited genes, the female also having sex limited genes, but the sex limited genes express only in female, because for the expression of sex limited genes in the female what are present? Hormones are present, but in our body hormones are absent for the expression of sex limited genes. So in our body the milk producing genes are not expressed. In order to assess the sex limited genes in the male parent, we observe the offspring. Based on offspring we have to assess the sex limited genes of male. Such type of test we call it as progeny test. Here what is meant by progeny? Offspring. By observing the offspring we can assess sex limited genes of male parent. So the answer is, the next one is assertion. Hemophilia, a sex linked character, is more common in man than woman. Now as we observe the hemophilia, that is a genetical disorder. The gene responsible for the hemophilia is recessive. It is present on which chromosome? X chromosome. Now as we observe the male, this male person has no hemophilia. In the brackets into mark indicates he has no hemophilia. So what is the genotype? XY capital H. Now a male has hemophilia. Now in brackets this indicates the male suffering from hemophilia. What is the genotype? On X chromosome small h. Now as we observe the male animal, how many X chromosomes? One X chromosome. The gene for the hemophilia is recessive, present on X chromosome. So sex linked recessive genes more expressed in male, because in the male how many X chromosomes are present? Only one. So the assertion is correct. Reason: a single recessive gene in men but two genes in woman are required to express the sex linked recessive character. In order to express sex linked recessive character, that recessive gene present on both X chromosomes in female. For example, we go for the female. Now one condition is the female has no hemophilia. XX capital H capital H. Second condition, the female has no hemophilia but carries that gene, capital H, small h. A female has been suffering from hemophilia. Now, such a recessive gene present on both X chromosomes. If recessive gene present on only one X chromosome, the disease is not expressed in the female, because for the expression of recessive gene, that must be present on two X chromosomes in case of the female. But in case of male, how many X chromosomes? One. So, sex-linked recessive genes express more in males than females. So, both are correct, or he explains A. The next question. Read the following statements. Sex-influenced genes are dominant in one sex and recessive in the other. Now we go for the sex-influenced genes. Sex-influenced genes, they are present on autosomes, and these are present in both male and female. Okay? Sex-influenced genes more express in male, less express in female. What is the best example? Baldness. What is the best example? Baldness. That we call it as Battatala. Now as we observe the genotype, capital B, capital B, capital B, small b, small b, small b. This is male animal, female animal. Now, if capital B genotype present, male and female having the baldness. If the genotype capital B, small b, the male has only baldness, female has no. If the genotype small b, in male and female no baldness. So, as we observe the male, in the homozygous dominant, in the heterozygous condition, both conditions expressed in male. So, sexually influenced genes more expressed in male, less expressed in female. So, here the first statement, sexually influenced genes are dominant in one sex, that is male, and recessive in other, that is the female. Now, second statement, sex limited characters, already we discussed. Sex limited genes present in male and female, but they express only in female, because for the expression of sex limited genes, what are required? Hormones. They are present only in the females. Now, first statement, second statement correct. We go for the third. Pseudo autosomal genes are called completely sex linked. No, they are called incompletely sex linked. So, the third statement is wrong. Fourth one, Y linked genes. Where Y chromosome present? Only in the father. The Y chromosome always passes from father to son, but not to the daughter. So, here Y linked genes sometimes found in the females, that is wrong. Y chromosome always passes from father to son, but not the daughter. So, here what are the correct statements? Now, the next question. In the Ian Wilmot's cloning experiment, the cloned dolly resembles, already we discussed, from the surrogate mother. After gestation period, the cloning animal came out, that was named by Ian Wilmot as the dolly. The dolly resembles the first female sheep, because from the first female sheep he has taken vegetative cell. In that vegetative cell nucleus present. From the second female sheep he has taken ovum, but from the ovum he removed the nucleus. So, the dolly resembles the female sheep from which we have taken the udder cell. So, here in the Ian Wilmot's experiment, the cloned dolly resembles udder cell donor. So, the answer is. Next question. Testis determining factor gene. Now as we observe the Y chromosome, the genes present on Y chromosome are haploid. They are also called hemizygous. They are also called Hollandric. Okay. Always Y chromosome passes from father to son only. Now an example is hypertrichosis. And the second one is testis determining factor. Third one is porcupine skin and webbed toes. Now, webbed toes, it is not a disease. As we observe our legs, each leg has toes. In some human beings, between toes, these are the fingers but not the toes. Regarding the legs, between the toes, the skin expands. That we call it as webbed toes. It is not a disease, just it is a character. Porcupine skins. As we observe our skin, on the skin the hairs are present. The hairs are soft. The one who has been suffering from the porcupine skin, in his body, the hairs are spine-like. That is a disease. Testis determining factor. For the development of testis, the gene is required. That is present on Y chromosome. What about the hypertrichosis? The hairs present on external ear. My father has hypertrichosis. In my father body, on the external ear, hairs are present. Now you observe, I am also having the hairs on my external ear. It is hypertrichosis. It is a character, but not a disease. So, for all these characters, the genes present on Y chromosome. Now here, testis determining factor gene present on which chromosome? Y chromosome. So, the answer is. Now the next question. Incompletely sex-linked genes are incompletely sex-linked genes. Now as we observe, this one we call it as the X chromosome. Okay, for our kind information to explain the concept. Now this is the X chromosome and this is the Y chromosome. The X is large, the Y is small. Now, on the X chromosome, the genes are present. The genes present on X chromosome are haploid, no doubt. Now, on the Y chromosome also, the genes are present called haploid. X and Y chromosomes are separate, so called non-homologous. The genes present on X and Y chromosome, they do not undergo crossing over. They do not undergo crossing over. The genes present on X and Y do not undergo crossing over because they are non-homologous. They are separate. But here what we are asking? Incompletely sex-linked genes. As we observe the X and Y chromosome, here and there some homologous portions are present. This is homologous portions. On these homologous portions, the genes are present. Now you observe the diagram. The genes present on homologous portions of X and Y, but not on X and but not on Y. So these genes are called incompletely linked genes or pseudo-autosomal genes or XY linked genes, so that these genes express equally in male and female. Now, incompletely sex-linked genes confined to only males, confined to only females, present in both the sex. So the answer is present in both the sex. Next question. List 1. Beadle and Tatum. They proposed one gene, one enzyme. That is the old concept. One gene does not produce the entire enzyme. One gene, one enzyme hypothesis proposed by Beadle and Tatum. What about the Griffith? He conducted transformation experiments. What about the Benger? According to the Benger, as we observe the gene, G means gene. What is the gene? It is the part of DNA. According to the Benger, the part of the gene produces one polypeptide chain. That gene is called cistron. The part of gene undergoes mutation called mutant. The part of gene undergoes recombination is called recon. Mutant, smallest. Recon, also smallest. The smallest one undergoes mutation, mutant. The smallest one undergoes recombination, recon. The one that produces polypeptide chain called the cistron. This concept was explained by Benger. What about the Morgan? The sex-linked inheritance or sex linkage explained by Morgan. Now the answer is, next question. Non-coding regions of heterogeneous nuclear RNA. Now as we observe the eukaryotes, in the eukaryotes, this one is heterogeneous nuclear RNA. What is the direction? 5 dash, 3 dash. Now in this RNA, exons, introns, exons, introns, exons, introns, and goes on. Now the exon responsible for the transcription, translation. It gives mRNA, that gives protein. And so it is responsible for the coding. What about the intron? It is responsible for the only transcription. It gives only mRNA, that does not give the protein. So intron is non-coding. So here what are removed? Introns are removed. The introns removed by what type of proteins? Spliceosomes or spliceosomes. Here you have to observe non-coding regions of heterogeneous nuclear RNA removed by what type of proteins? Spliceosomes. They are present in the DNA strand. Such a process we call it as RNA splicing. Now the answer is, next question. A repressor protein of lac operon transcribed from. Now as we observe the gene expression and regulation in prokaryotes, now this is the DNA strand. Okay. Now in the DNA strand, what are the structural genes? A, Y, Z. These are structural genes. On the left side of A, Y. On the left side of Y, Z. On the left side of Z, what is present? Operator. On the left side of operator, what is present? Promoter. And on the left side of promoter, what is present? Inhibitor or regulator. Now the regulator gene gives rise to what type of protein? Repressor. Okay. Now the repressor binds to operator. Now you observe the diagram. Repressor is a protein. It is attached to operator. As long as the repressor present, the operator does not work. Operator off. Whenever operator off, the structural genes do not work. They are also off. Now here, a repressor protein of lac operon is transcribed from, this is I, inhibitor gene or regulator gene. So the answer is, next question. the cross between white leghorn and Rhode Island red. The cross between white leghorn and Rhode Island red. This is Mediterranean variety. This is American variety. No doubt, this is efficient egg layer. Now, the female white leghorn crossed with the male Rhode Island red. And this is one breed. Okay, one variety. This is another breed, another variety. Cross between the two breeds, we call it as cross breeding. And the result is called crossbreed. Now you observe the cross between the white leghorn and Rhode Island red is. Next question. Sex-linked recessive characters follow skip generation inheritance. We are asking sex-linked recessive genes. Now, this one we call it as the male and this one we call it as the female. Now we are discussing the Drosophila. Okay. Now, this is the male parent. This is the female parent. The male has white eye color. You observe the cross. The female is red eye color. Here white eye, a disease, red eye, no disease. Now what is the genotype? XY on X small r. Genotype XX capital R capital R. Now we go for the cross. XX capital R small r, this is female. What is the color? Red color. Now XX capital R small r, female, red. And XY on X capital R, red. This is the male. Now XY, this is the male, red color on X capital R. So in the F1 generation all are red. Okay, female, female, or daughter, daughter, son, son. Okay, here you observe the daughter is carrier. She is carrying the gene for the white eye color. Okay, here the daughter is carrier or heterozygous. Now, you take any two. Now you go for this one. We are getting X R, X small r. We are getting X R. Y. Go for the cross. XX capital R capital R. This is the female, red eye. And this is XX capital R small r. Female, red eye. Now you go for XY. This is male, red eye. Okay, now you go for XY small r. Male, white eye. Now in this cross, this one is considered as the grandfather. Now here, this one we considered as carrier daughter. And this one we considered as the grandson. Now, from the grandfather, the white eye color jumped to grandson through the carrier daughter. Such jumping we call it as skip generation. Such skip generation can be explained with reference to X-linked recessive genes only. Now the answer is both are correct or he explains CA. Next question. The genes that do not undergo pairing or crossing over during the meiosis in males. Now as we observe the male animal. Now in the male animal, X chromosome, Y chromosome. This is X chromosome and this is Y chromosome. Now on X chromosome the genes are present, on Y chromosome genes are present. X, Y both are separate, non-homologous, already discussed. So these two are non-homologous. So the genes present on non-homologous chromosomes do not undergo crossing over. So here the genes that do not undergo pairing or crossing over during meiosis in males, X-linked genes as well as Y-linked genes. So the answer is 1 and 2. The next one. Find the wrongly matched. Paul Meiter and Brinter, they produced first transgenic animals. Beadle and Tatum, they proposed one gene, one enzyme hypothesis, but not one gene, one polypeptide. That was proposed by Ingram. Jacob and Monod explained expression and regulation of genes in prokaryotes. That is called operon synthesis. Sutton and Baveri explained chromosomal theory. So the wrong answer is the next one. Hypertrichosis, already discussed. Hypertrichosis, the presence of hairs on external ear. The gene for this character present on Y chromosome. Y chromosome always passes from father to son, but not the daughter. So the characters, the genes for the characters present on Y chromosome always passes from father to son. The genes on Y chromosome are called Hollandric. So hypertrichosis is present only on men. The genes for the hypertrichosis is present on Y chromosome. So the answer is next one. Inducer in lac operon of E. coli. Now in the lac operon concept, Z, Y, A, operator, promoter and inhibitor. Now the inhibitor gene produces a repressor gene. The repressor binds to operator. So the operator becomes off. As long as repressor protein present, it does not work. If this does not work, this does not work. So we want to remove repressor protein. How to remove? The repressor removed by what type of material? Inducer. Here what is the inducer? Lactose. If there is no lactose, yellow lactose works as a lactose. If the inducer, whenever the inducer attaches to repressor, the repressor removes. Automatically operator on. Automatically these ZYA genes on. Now you observe the question. Inducer in the lac operon of E. coli. The answer is lactose. If there is no lactose, yellow lactose works. It is isomer of lactose. So the answer is next question. The part of DNA chain responsible for the synthesis of one polypeptide according to Benger, cistron, mutant, recon. Part of the DNA that synthesizes one polypeptide chain, that we call it as cistron. The answer is next one. The template strand for the transcription into mRNA in DNA and direction of transcription is. Now as we observe, this is the DNA strand 5' 3', 3' 5'. In this strand, information present ATGC. In this strand, quite complementary, TACG. Now this is sense strand. This is also sense strand. Now, at the time of formation of mRNA, this one acts as template. Okay. Now, This strand by a process called transcription gives rise to mRNA. It will be synthesized in which direction? 5-3-. So it is complementary to this one. From this template, this synthesizes. So on this strand, there is no information. The information of this template transferred to mRNA in the form of the codons. Okay. Now, as we observe the template 3-5-, here there is no information. It is called antisense. Because on this strand the information transferred to mRNA. So, what is the direction of template? 3-5-. What is the direction of mRNA? 5-3-. Here we are asking the template strand for the transcription into mRNA in DNA, and direction is, the answer is antisense strand 3-5-. From this, what will synthesize the mRNA in which direction? 5-3-. Now the answer is this one. Now the next one. When the S strain, that is related to Diplococcus, was treated with DNase enzyme, transformation did not occur because the DNase enzyme digests the material of S strain. DNase enzyme digests only DNA material. So whenever the material of S strain treated with DNase, there is no transformation, because in the S strain the material completely digested by DNase. So there is no transformation. It means in the S strain, what type of material is present? The DNA. Such DNA synthesized by DNase enzyme. So there is no transformation. The answer is next one. Number of alleles. Now regarding the blood groups, number of alleles IA, IB, IO. Number of alleles 3, and number of phenotypes A blood group, B blood group, AB blood group, O, and number of genotypes IA, 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 IO, IB, 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 IO, IA, IB, IO, IO, 1, 2, 3, 4, 5, 6, 4, 3. Now the answer is the next one. Anti-D. Now this one is called REH antigen. The REH antigen identified in rhesus monkey. The REH antigen is also called D antigen. It is the dominant. Against REH antigen, antibody present, that is called IgG. It is called D. Now you observe, anti-D antibodies are, what type of antibodies? IgG. Okay, the answer is the next one. In human beings, Klinefelter's syndrome, that is a disease, occurs only in males. That person is a sterile male. In that male, femaleness is not completely suppressed, because as we observe the Klinefelter's male, now the male animal, in the male animal AAXY. Now because of the non-disjunction, the sperm has AXY. Now this sperm crossed with this ovum AX. Now you observe AA, one set of A value 22, another set of A value 22, so 44. X, X, Y. Here 1x is coming from the ovum. So The one who has been suffering from the Klinefelter's syndrome, in his body one extra X present, that is derived from the female. So in his body the female characters are not completely suppressed. So here the answer is the next one. Environment favors sex determination in, as we observe the Bonilla viridis, this is female Bonilla viridis. This one we call it as proboscis. This is the male Bonilla viridis. This is the female and this is the male. The male religious sperms, the female religious ova. Both undergo fertilization, we are getting the zygote. The zygotes are hatched out into larvae. Now some larvae freely swims in marine water. Freely swimming larvae will become the female. Some larvae attached to proboscis of female. Those larvae attached to proboscis of female will become the male. That means here what is the environment? The marine water. The larvae freely move in the marine water will become the female. So environment favors sex determination in Bonilla viridis. Next one. According to Crew, the male suppressing the bird ovary can. Now as we observe the hen. Now as we observe the hen. In the hen body, testes and ovary both are present. As long as ovary present, the testis is not working. Whenever the ovary is removed, automatically the testis is functional. Such hen acts as the cock. Okay, now according to the Crew, the male suppressing the bird ovary can prevent the development of testes. As long as ovary present in the hen body, the testis is not working. If ovary is removed, automatically the testis is functional. Then the hen acts as cock. The last but one question. Bar body is said to be associated with, in case of female human beings, 2 X chromosomes are present. Now in 1 X chromosome, here and there the chromatin material, here and there the chromatin material is condensed. That we call it as bar body. On 2 X chromosomes, on 1 X chromosome bar body present. Such X chromosome does not functional. So here bar body is said to be associated with only on X chromosome. Last one. In which animal the sex of the offspring determined by the number of sets of chromosomes? As we observe the honeybee family, in queen body diploid set of chromosomes 32. In case of male honeybee, haploid set of chromosomes 16. So in which animal the sex of the offspring determined by the number of sets of chromosomes? In queen 2 sets diploid. In drone, that means male, 1 set haploid. So the answer is honeybee. Students, in the next class we have to discuss the human genome project and the evolution part.